Focus, Dr. Kowinski, focus. All right. So at the very, what was that, Elizabeth? Oh, you didn't. Okay. At the end of class last time, we were talking about this Trivers and Willard model that looked at the quality of the mothers and how the offspring that they produce varies with the quality of the mother. But the fitness gains of male offspring versus female offspring changes with maternal quality, but the nature of that increase in fitness varies between sons and daughters. So daughters increase in quality at a gradual clip and it's linear, whereas son reproductive success increases exponentially with mother quality, such that above a certain level of maternal quality, mothers should produce more sons than daughters, and below that they should produce more daughters than sons. And then we illustrated this with the whole red deer example, showing that the lifetime reproductive success of daughters doesn't really change at all across maternal quality, whereas the lifetime reproductive success of males increases dramatically with maternal quality, which means that we should see females in these areas of dominance within the harem should be producing more males than females, because these males are going to have much higher reproductive success over the course of their lifetime than if you produce males, if you're a low quality female, in which case you should be producing more females than males. This Trivers-Willard model then becomes a general way of thinking about how different aspects of the organisms that are influencing sex allocation, do it. So what changes in each of the next couple of sections of the book is actually just what is plotted on the x-axis. In this case, it's the dominance of the females in these harems, or maternal quality in general. But then as we move from one thing to the next thing, it's this same graph just over and over and over again, and we just change the x-axis. So keep that in mind as we move forward through the rest of this chapter. So you can also think about mate attractiveness. So in this case, we have blue tits. This is this cute little bird. It has this blue area on the top of its head. That area also reflects ultraviolet light, and the birds can perceive this. And as it turns out, the level of reflectance of that turns out to be a good indicator of male quality, whether it is communicating parasite resistance or what. Higher quality males that are more attractive to females, generally have higher levels of UV reflectance in this patch of feathers. And so you can measure that UV reflectance, and what they have found is that when a female mates with a male that has high UV reflectance, she tends to produce more males. So she produces a skewed sex ratio, skewed towards males. And when she's mating with a male with a low UV reflectance in that area, she tends to produce a sex ratio that is skewed more towards females. Presumably, the reason that she's doing this is that if you're mating with a male of high quality, that mating, if you produce sons out of it, those sons will also carry those same traits. So if you have a high quality male that you're mating with and you produce a bunch of sons, all of those sons will have the same traits as that male. And as a result, they will also have good mating success because they possess those traits that they inherited from their father. If, on the other hand, you're mating with a low quality male, to invest in sons doesn't really gain you anything, because those sons are going to have a hard time finding mates, and so then you skew the sex ratio towards females, because the reproductive success of females isn't determined by that trait that is the trait in the males. And so all we've really done is we've changed what is on the x-axis here, so that now rather than maternal quality, it's now paternal quality. And once again, you see that the sex ratio changes because the success of males increases with the success of sons increases with the quality of the male parent in this case. And I have misspelling on the slide. They apply the same Shivers and Willard idea to environmental sex determination. So somebody remind me what we mean when we talk about, there's lots of misspellings on this slide. Good grief. What's an example of environmental sex determination that we've talked about in the past?
The temperature, like the turtles.
Okay, temperature-dependent sex determination in turtles. So in environmental sex determination, it's a situation in which the environment in which an individual goes through early development affects what sex you are. And the only way that this works, according to the Sharnov-Bull model, is if the environment that you develop in has different consequences for males and females. So let's think about temperature-dependent sex determination as an example of this. And so if we look at snapping turtles, what you observe if you go out and measure the incubation temperatures of nests that are on the cool side tend to produce all females. Is that right? This is the proportion of males. Males divided by the total. Proportion of males is 0. This is all females. Then you produce males in the middle, and then you produce all females at the high end. And so what Sharnov and Bull's model essentially says is that you produce females at this temperature because females at this temperature have higher fitness than males that are produced at this temperature. You produce males at this temperature because males at these temperatures have higher fitness than females do. And you would produce females at these temperatures because females have higher fitness than males at these temperatures. So what I want to do is basically just zero in on one little section of this graph. This graph represents a situation where you go from, presumably, all females to mostly females, very few males, an even distribution of males to females, to mostly males, very few females, to a point that you are finally at nothing but males. As you go across this narrow band of 8 or 9 degrees of temperature. So assuming that I made my graph for the next slide correctly, I didn't make my graph for the next slide correctly. I did it just the opposite. What you should see, so flip this around in your head. Red is female fitness and blue is male fitness. So I labeled my things wrong. What you should see is as you go across this range of temperatures, you should see that female fitness is highest at those temperatures that tend to produce females, and it declines as you move into that middle temperature range. And as female fitness is declining, male fitness is increasing. And so once again, in this case, what you're really just seeing is instead of maternal quality or paternal quality being on the x-axis, you're essentially trading that axis out for temperature, such that as temperature increases, male mating success, male fitness goes up, while in this case, female fitness is actually going down. So rather than that figure looking kind of like this, where this is for males and this is for females, you have a situation that's just slightly altered in that you have where this is for females. in which case an individual should produce females on this side of whatever that threshold is, and you should produce males on this side of whatever that threshold is, and that threshold occurs in this area here at around 20 to 21 degrees centigrade. Now, this is tricky to test. And one of the reasons that it's tricky to test is because it's very difficult to maintain eggs in an incubator in a lab at these really narrow bands of temperature, so that you would get mixtures of males and females at these temperatures where presumably fitness would be suboptimal. So in order to test this, you have to go out into the field and you have to get lots of turtle eggs from lots of females, and you have to get them early enough that they haven't gone through that period where sex determination occurs. So remember that there's this narrow period during which sex determination happens. Then you have to divide these up. Presumably you're getting lots of turtle eggs because you're getting lots of turtle eggs from lots of females. And then you have to have incubators, and you have to have an incubator that's set at 16 degrees, an incubator that's set at 21 degrees, an incubator that is set at 22 degrees, and an incubator that is set at 24 degrees. And that covers this range, going from nothing but females to nothing but males. So what you would expect to see is nothing but females here, nothing but females here, a mixture of females and males here, a mixture of females and males here, and a mixture of and nothing but males here. And in this case, what we should see is we should see females doing better when they come out of this incubator and males doing better when they come out of this incubator. So what you do is you incubate the eggs, you get the sex ratios that you get, and then you cut those individuals loose out into the world, and then hopefully you then measure some correlate of fitness. What measure of fitness did we use in the red deer situation? What does LRS stand for? Lifetime reproductive success. How long do turtles live? Elizabeth? They live a long time. Because they live a long time, how long does it take them to reach maturity? It takes a long time, 18, 20 years for a snapping turtle to get to the size that it's mature. So when you then go to all this trouble to hatch out all these turtle eggs, and you get them to come out at the right sex ratios given the temperatures that they're at, you then have to measure fitness, but the fitness that you're measuring is not going to be lifetime reproductive success, because you as a biologist just don't live that long. And so you have to do something else. What other kinds of things could you measure that might be correlates of fitness? These turtles are small when they come out, so they're on the tiny side. You might measure growth rate. Maybe give everybody the same amount of food and see who grows faster. That would be a good idea. That's something that you could measure over a reasonable time period. And presumably, if you're growing faster, you reach the size at which you reach maturity faster, and so that might actually increase your lifetime reproductive success, simply because you now have more years of reproductive lifespan to take advantage of. So that would be a correlate of fitness, even though it's not fitness directly. What would be something else that you could measure?
I mean, this is more like, I don't even know if it would work with turtles or whatever, but it's like, you could somehow share, like, if one's more dominant than other ones,
You could maybe do that.
resources and some of that.
I don't know anything about that, but that's a possibility. What else could you measure that's maybe more directly related to fitness? You just test whether they live or not. Cut them loose in the environment, put them in a pond, come back a year later, collect all the turtles from the pond, and see who lived and see who died. You can mark turtles. You can either implant them with pit tags like you put inside of pets. They're little transponders that have a unique number associated with them. Pit tags are really easy to put into turtles. They handle that really well. So you could actually just cut them loose, leave them out in nature for a year, and then come back a year later and see who survived or not. If you did that, you might see that these females fared pretty well. These females fared pretty well. These males didn't. These males fared pretty well. These males fared really well, and these females didn't. If that was the case, then you would take this as evidence that the Charnoff-Bull model of thinking about um, temperature-dependent sex determination works. And you could do this at this end of the spectrum. You could also do it at the high end of the spectrum. It doesn't really matter. The predictions would be just the same. They would just be flipped because you would have females here at high temperatures and males at low temperatures. So this is how you would test it, but going through this process of thinking, well, how would we do this with snapping turtles? You can see why two things have happened. One, there haven't been very many experimental tests done of this idea that Charnoff and Bull have, and many of the tests that have been done have not been done particularly well, such that the results are kind of ambiguous. And so this remains to be one part of sex allocation theory that is not particularly well tested and could be tested better perhaps in other species. Your book talks about a species of freshwater shrimp where this has been tested in a more rigorous sort of way, but outside of that, with vertebrates at least, it hasn't been tested adequately, even all these years later. Charnoff and Bull put their model out in 1977, and there still hasn't been a great example with the kind of animals that typically do this. This is found fairly commonly within reptiles. So alligators and crocodiles experience temperature-dependent sex determination. Some species of geckos experience temperature-dependent sex determination. A lot of turtle species experience it. So in the vertebrate groups where this is found, oftentimes the difficulties of working with those groups is just really makes it difficult to test this idea in some of the more common species that we have that do this. So sex change is another thing that organisms do. And once again, we're going to still think about this Trivers-Willard model, but sex change is oftentimes associated with body size. And so now, rather than maternal quality being on the x-axis or paternal quality being on the x-axis, now body size is going to be on the x-axis. So sex change is a situation where individuals mature into one sex and then at some time later they change sexes. So you might mature as a female, and then as you continue to grow, you then change sex to a male. And this basically happens because fitness changes with either age or size for a particular sex. So imagine that you are in a species that is polygynous, and it is resource defense polygyny. What are some characteristics that you know from our now extensive conversations about mating systems and things like this? What are the kinds of things that you would expect in resource defense polygyny from the standpoint of characteristics of males and characteristics of females? Resource or female defense polygyny, either one. What is polygyny for starters? One male, multiple females. If you have all these females, what's going to happen to you? You're going to have high fitness, but what else happens to you? Well, that's high fitness, lots of offspring, sure. Is that just free or does that come at a cost? Okay, so typically in polygyny, you don't provide parental care, but you do have to do something else. Resource defense or female defense polygyny. Why do I call it resource defense or female defense? You have something that females want that make you be able to be polygynous. So you got something that they want. You also have something that other males want. So now some bigger, burlier male comes along, sees your group of females and your resource and says, what? That's mine. And so you have to defend your territory against this interloping male. And in this case, chances are the bigger, stronger male prevails in that competition, takes over your harem of females, and now he's got all the fitness, and you are on the outside, not knowing what to do with yourself because you are a small male trying to maintain a group of females, trying to maintain a territory that has resources that females want. In this case, as you grow up and mature, would you want to mature into a male or would you want to mature into a female? Hmm? Why? Yeah, in this case. Yeah, so if you mature first into a female, you're going to start off as a small female. Small females will always find male mates. Why? Males are able to mate with multiple females, and so they'll mate with any female. The female has a valuable resource that the male needs for reproduction. The female has eggs. And so small females are going to find mates. Now, a small female may produce fewer offspring than a large female does. Large female may produce more offspring, but once again, think about the Trivers-Willard way of thinking about things. If we think about size from small adults to large adults, what we would see in terms of reproductive success, whether it's lifetime reproductive success or a single season of reproductive success, is small females produce few offspring, so this is 0 to a lot of offspring. As females get bigger, their reproductive success may get bigger simply because they have bigger bodies, they can fit more eggs in there, they have more energy perhaps, and so this increases gradually. But when we look at males, there's some threshold size at which point males can maintain and defend a territory. So below that size, males have very low reproductive success, but above that size, their reproductive success increases. So once again, this Trivers-Willard model of thinking about things, it's just replacing maternal quality for size. And at this point right here, this is when you should make the shift from being a female at these sizes and being a male at these sizes. Simply because, in this case, both sexes are probably increasing in reproductive success as they get bigger, but the shape of the curve is different for males than it is for females. And so at that point, you should shift. There might be other situations where maybe the size of the female influences egg production to the point that the female experiences kind of exponential growth in egg production as she gets bigger. In that case, if male fitness increases At a slower rate, what would the outcome be for sex changing? Should you mature as a female or as a male? No, it would matter. But what would it be? At low size, oh, sorry, this is the male curve. This is the female curve. Yeah, you should mature as a male because you have higher fitness at small sizes as a male, but because the increase in female fitness, because she just has a bigger body and as a result can produce more eggs, increases faster than the increases that accrue to males. And so once you cross this threshold here, you should now be female at larger size and you should be male at smaller size. So it all depends upon the specifics of the situation, but still it's just a matter of how quickly does fitness increase with increasing size for males and females. That's all that really matters. An example from your book is the example of clownfish, these anemone-dwelling fish. What's peculiar about anemone-dwelling fish? What does the male do? Male does parental care, guards the eggs in the anemone. What do you know about anemones? They're not very big. What? Let's not think about emotional responses of Nemo. Let's think about the biology of Nemo, because the biology of Nemo is so the anemone fishes actually they don't get stung by the anemones because they have a protective coating that basically tells the anemone that they're not a fish. Essentially, it tells them that they're anemone. And so that's why they can live in the anemones, but oftentimes the anemones can only hold one male and one female. So there's just not enough space in the anemone for more than one male or one female. So in this case, what does it behoove a male to be? It behooves a male to be small. And so in this case, in anemone fishes, which do change sexes, you tend to find males mature, these species mature as males at small size, and as they get bigger, they, if they are females, will have higher reproductive success because they can produce more eggs. And so there comes a point where they reach a threshold size, at which point they transition from being males to being females because of the size limitation of the anemone that you occupy. Once again, it's just Trivers and Willard over again. Same idea, same thought process. Just in this case, age or size replaces maternal condition. So we have words for whether you mature as a female or you mature as a male. Protogynous is female first. Protoandrous is male first. Example of female first is the blue-headed wrasse. Once again, this sex changing occurs a lot in fishes, as it turns out. So most of the examples in your book are fish examples. You mature as a female first because, once again, males have to maintain territories to attract females, and you're much more likely to maintain that territory if you are a large male. So females at small size, males at large size. And also females tend to be choosier and they tend to choose larger males. So for those reasons, males tend to be larger, older individuals and females tend to be younger, smaller individuals. In the case of protandry, we have male first. This is a situation where males benefit from small size and mating. This happens in the clownfish, which I've already talked about. And the main reason for this is the size limitation of the anemones where they hang out and brood their young. So once again, it's not set in stone. You have to think about the biology of the organism to know what the nature of the sex change is going to be, whether it's going to be female first or male first. Oh, I already drew this on the board, so we don't really have to think about this.
So biologically, do they undergo the change or they grow like more with like good organs?
They actually, as far as I know, they actually grow new organs. It is pretty crazy. I guess so. I don't know that much has been done on gene expression. Um, One of the things that I've learned over the course of teaching here for 22 years and trying to keep up with literature is gene expression has as much to do with phenotype as the actual allelic expression. So we talked about this earlier in the semester. When you think about the genetics that influence the phenotype, you can have genetics that influence phenotype in that I have the dominant allele for tongue rolling, so I can do this. But if that gene for tongue rolling isn't being expressed, I wouldn't be able to roll my tongue. And so the way I learned genetics when I was in school is like allelic variation is everything. You have a gene, it gets expressed, and that makes you who you are. We don't really think that anymore because we know more about how genes are regulated. And so I don't know what gene regulation is in these sex-changing fish. That would be a really interesting thing to go and look at. I should make that a literature review for future generations of students. What's that?
Good luck to them.
So, but what I do know is I know some other gene expression studies that have been done that are equally striking. So let me give you an example just to illustrate why this would be possible just through changes in gene expression. You guys know about pythons and boas. So if we think about foraging behavior, a python, let's say you have a reticulated python, 36 feet long, big, huge thing in the middle. These things take down large hooved mammals and eat them. And so what they'll do is they will kill a large animal and they will swallow it whole. So now you have a snake with this gigantic bulge in the middle of it, and it takes some time for the snake to digest that thing. But then after that prey item is digested, that snake might not eat for another 6 months. So the snake eats a gigantic meal and then doesn't eat anything else for 6 months. Well, this is a curious kind of way of going about foraging, and it's always been a curiosity and a source of interest for biologists. And there's a biologist at University of Texas at Arlington who is a herpetologist and also does genetic work. And he has been interested in what happens in the gut of these large carnivores that eat these big meals and then go long periods of time without eating. Well, what he found is that during the time between meals, they basically let their digestive system atrophy. So your digestive system has muscles that go around it, connective tissue, and it has an endothelial lining. And your endothelial lining produces enzymes and acids, et cetera, and you maintain that. And it's constantly being sloughed off, as all epithelial tissue is. All epithelial tissue loses cells and has to be regenerated. Well, what snakes do is in between these meals, they just let that degrade. Just goes away. They don't have an endothelial lining of their gut. Then they eat a big meal, and all of a sudden, they rebuild all of that lining of their gut. It rebuilds, it processes that prey item, and once that prey item is digested and that snake poops it out, the digestive system just atrophies again. And when you look at what's happening in the gut, if you do an RNA-seq analysis of the cells in the gut, what's happening is they basically turn off all of the genes that are responsible for maintaining the gut. And then when it's time to eat again, they turn all those genes back on. They rebuild the gut. And then after that's done, they get rid of it. And what they do is they save a ton of energy because they're not having to maintain that system that's not being used. And so they have an adaptation that conserves energy And it evidently is costly enough energetically to maintain a gut that's not being used for anything, that they've just evolved a way of atrophying it. And it's all done by gene expression. So if you look at his papers, they have heat maps of gene expression when they're feeding and when they're not feeding. And it's like night and day in terms of gene expression. So gene expression can be really, really important. And so I don't know of a paper that's been done on clownfish or any of these sex-changing fishes, but my guess is it's just all about gene expression, if I had to guess. But if somebody hasn't done that yet, somebody needs to do that. That would be a fascinating study to do. Oh, I'm going backwards. I need to go forwards. The last topic in your book on this particular issue is sex ratio disorders. And this is an interesting situation in that it involves interactions among species. So there is a type of bacteria called Wolbachia, and these are bacteria that essentially infect and affect insects for the most part. And what they do is that these bacteria kill off male offspring. And so they're inside the females, and they basically infect the male offspring and prevent them from being hatched. And as a result, it skews the sex ratio of the insect that it infects. And so if you have this in a butterfly species, what happens is that the butterfly species produces many more females than males. So we have a female bias sex ratio. And the reason that the bacterium kills off the male offspring is because the Wolbachia is only transmitted through the females. It doesn't transmit through males. And so the males are kind of, if you find yourself inside of a male, you are kind of at an evolutionary dead end. And so because of this, Wolbachia basically tries to get rid of as many male offspring as possible. That produces insects that produce a female bias sex ratio. But the insect species, in many cases, don't evolve mechanisms to counter the action of Wolbachia, because in many cases, having more females is actually beneficial to the females of the species that are infected by Wolbachia, because they're reducing competition from other males. And so once again, as long as there are enough males to reproduce with, you don't really need males around for anything else. And so it all depends upon, once again, the specifics of the situation. Some butterfly species have evolved mechanisms to counter the action of Wolbachia, but many of them haven't, simply because there's no cost in having a female biased sex ratio. There are a number of people around the country that study Wolbachia and kind of what the evolutionary consequences of Wolbachia are. This is something that has been known for a while, but people are still in the process of trying to figure out all the details of how this coevolution works between a bacterial species and insect species. And I think that is all for.
Is that transmitted to the female offspring as well?
Yeah. Yep. Yep. So it just goes female to female to female to female. So that's why there's no incentive for Wolbachia to keep males around. We will start the chapter on altruism and spite next week, so read that over the weekend. Altruism and spite serves as kind of your book author's introduction into social behavior more broadly. But so they have a chapter on altruism and spite, and then they have a whole chapter on social behavior, which we will not get to because we're running out of the semester. But that begins to set the stage. Altruism and spite are kind of two extremes of social behavior, so they use that as an introduction to it. So you'll learn some things about social behavior, but you won't learn everything about social behavior. But altruism and spite can be kind of fun, partly because it's easy to think about it in humans, and it's also easy to think about it in other organisms as well. Um, I'm sure you've seen examples of presumed altruism in humans, but you can also see similar examples in the animal world outside of humans as well. And so that's what we'll talk about Monday and Wednesday. Well, really Monday of next week, because Wednesday, Emily will be here to talk about mating systems in frogs. How's that paper coming for you? Okay, I know what that means.
Yeah.
Um, there's a section in the discussion where they talk about the mating system being simultaneous or serial polygyny and polyandry. And you might find that confusing. It's polygyny and it's polyandry. But the key to understanding that is actually downloading the appendix. So it makes reference numerous times throughout the results and in the discussion to the appendix. If you go to the appendix, do I still have it up on my computer? All right, let's just do this. So in the appendix, it has each female, and it has which male they interacted with, and whether that interaction led to a mating, or whether the interaction was cut off by the male, or whether it was cut off by the female. And so, for example, female 1 mated with, or interacted with male 3, 1, 2, 3, 4 times, then interacted with male 8, then male 10, then male 8, male 8, male 3, male 8, male 8, male 10, male 3, male 3, male 3. So this individual interacted with male 3, male 8, and male 10. One female, multiple males, polyandry. So you can go through this table. They have a key at the bottom of this. Um, a heart is a successful mating. Yay, hearts. Courtship is a club. Courtship stopped by the females, club and diamond. Stopped by the males is club and spade. It represents tadpole transport. So this male provided tadpole transport. So by looking at this, this gives you some insight into the statements that are made in the result when they refer to females mated with anywhere from 1 to 3 males. When you look through the appendix, you see that the maximum number of males that females mated with were 3. And so this is useful and helpful to you. If you're not in the habit of downloading electronic supplemental information from journal articles, this is one of those journal articles where actually having access to it actually helps you understand the findings of the paper. I also don't always go and get the supplemental information, but in this case, it actually turns out to be quite useful. So you might find that helpful as you're going through and reading the article.
Where do you find that? Because this thing keeps taking me to MetaPress.
Go to, okay, so what's the name of the paper?
Parental investment potential reproductive rates and immune systems.
So if I copy this and go to the internet, go to the Springer link, So go to the link of the article there. You don't want to buy it because I've given it to you. But what you do want to do is you want to go down to electronic supplemental material, click on appendix, and you can get the appendix for free. And there it is. There's only one appendix, but once again, the appendix is actually useful in helping you sort through. It's a good idea. When I first read this paper, I found it really confusing the way they talked about things in the results. And so I just basically put the appendix in a printout next to me as I read the results and discussion. And as they talked about things, I went and found those frogs in the appendix. I'm like, oh, that's why you're saying X. And so I would advise you to do the same if you really want to understand it. All right. I will see you guys on Monday. Have a good weekend. Try and stay warm because